Today I'm going to be talking about the endomembrane system. It's basically a group of cell membrane organisms that are mutually interconnected, either by being directly continuous or with one another, or by sending one another vesicles. It provides compartmentalization, which allows different places in the cell to have different environments, and the organelles that are involved in the endomembrane system are the nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and plasma membranes. These are all found within a eukaryotic cell, which doesn't include the mitochondria and chloroplasts. The nuclear envelope. It's a double membrane similar to the cell membrane. It's a highly regulated membrane barrier as it encloses the nucleus to separate its contents from the cytoplasm. It contains a lot of number of proteins which have been implicated in chromatin organization and gene regulation, and there's a thin space between the two layers which connects the lumen of the rough endoplasmic reticulum. The Golgi apparatus is one of the first cell organelles. It's a distribution and shipping center for chemical processes in a cell. It consists of a separate flat membrane called the cisternae. It sorts out and transports products for various parts of the cell. It also modifies products by using the Golgi enzyme. The vesicle that pinches off the Golgi apparatus is moved to the cell membrane, can be sometimes called the lysosome. And together with the endoplasmic reticulum, the Golgi apparatus makes up the majority of the endomembrane system. Vacuole is a very important part of the endomembrane system. It's a membrane-bound sac. It's formed by pinching in of the plasma membrane. It transports products around the cell. Vacuoles carry out important functions like eliminating water, entrapping food and particles, and discharging waste. Endoplasmic reticulum, ER for short, is a membranous structure that forms a completely enclosed convoluted tubular sac. There's smooth ER and rough ER. Smooth ER is agranular endoplasmic reticulum and has no ribosomes attached, while rough is granular and has ribosomes attached. ER synthesizes activation and transport of macromolecules in the cell, however, depending on the addition of ribosomes or not. Smooth ER on the outer surface lacks ribosomes, contains enzymes important for the synthesis of lipids and steroids, detoxification of drugs and poisons, and metabolism of carbohydrates. Proteins targeted for transport to the Golgi apparatus are transferred from ribosomes on to the rough ER in the rough ER lumen, which serves as a site of protein folding modifications and assembly. Lysosomes are the primary components of intracellular digestion. They're membranous sacs of hydrolytic enzymes which are produced in the Golgi apparatus. Their main functions are just phagocytosis, which is in cell engulfs microorganisms, and autophagy, which is the cell recycling its own organic material. Lastly, we have the plasma membrane. It's a selective permeable barrier that surrounds the eukaryotic cell, forming a dynamic interference between the cytosol and the environment. And although it's not inside a cell, it's still included in the endomembrane system as it interacts with the organelles in the endomembrane system.